Hello, everyone, and thank you for tuning in to IFFGD's Research Award Winner Series, Where Are They Now? This program was designed not only to acknowledge this year as being IFFGD's 30th anniversary, but to also give us the chance to reconnect with some of our past Research Award winners. So today we actually have the pleasure of speaking with Van Oudenhove, who was our 2013 IFFGD Research Award winner for Junior Investigator in Clinical Science. Thank you so much for joining us today.
My pleasure. Thanks for having me.
Yes, so can you please tell us just a little bit about yourself, where you're currently located, and also your current research focus?
Sure. So I'm Lukas Van Oudenhove. I'm a psychiatrist by training, but I've been studying gut-brain interactions for my entire research career, basically, starting during my PhD back in the days 2004 to 2008, which was on brain processing of gastrointestinal pain in health and functional gastrointestinal disorders, and how psychological comorbidities was playing a role in that process of visceral pain perception. And since then, I've been continuing to study gut-brain interactions in humans, in health and disease. After my PhD, I did a postdoc on the topic, and then I became an assistant and associate professor. I'm running my own lab at the University of Leuven in Belgium, which is called the Laboratory for Brain-Gut Axis Studies. But currently, I'm located in the US. I'm on a sabbatical at Dartmouth College in an affective neuroscience lab here to learn more about advanced brain imaging techniques and apply these on the study of brain-gut interactions.
Yeah, no, thank you so much for that backstory and just a little bit more detail about what you're doing. It sounds very exciting. So we were talking about that you were the winner in 2013 for one of our IFFGD Research Awards. Can you explain to us just a little bit about how winning that award may have influenced your career?
Sure, yeah. It obviously came at a good time. I won in 2013, which was when I was just appointed as an assistant professor on a tenure track position. So I was just starting to become an independent researcher and starting my own lab. So, and it was one of the major awards I won in my career until now, even, which came at a very good time. So it certainly gave me a lot of credit and also a boost to get my own research group going. I'm certain that it helped me establish my own group. It gave me credit for obtaining funding in the area of functional GI disorders. And it certainly also helped to get tenured 5 years later. So I think it has made quite a big impact, and it came at a very good time. So I'm still very grateful for it.
Thank you. Thank you so much for sharing that with us. And I know you said that it kind of helped you kickstart the some of your own independent studies and research opportunities. So can you tell us a little bit about what current research topics you're working on?
So my lab is a human research lab. So I only work in humans. I don't do any animal research. But it is, I would say, quite mechanistic. It's certainly not purely clinical research in the narrower sense of the word. We study humans, both healthy volunteers and patients with disorders of brain-gut interaction, to look at fundamental mechanisms of gut-brain communication and how these may underpin symptom perception, gastrointestinal symptom perception in health and disease. But it also broadened towards how gastrointestinal hormones and other signals from the gastrointestinal tract may impact the brain and psychological processes in the brain. And recently, of course, we also started focusing on the gut microbiota, and particularly on mechanisms through which the gut microbiota can impact the brain and the emotional brain and psychological processes emerging from it in particular. So that's, in a nutshell, what my lab is doing.
Yeah, and definitely all current topics that are very exciting, and really a lot of research are going into both of those areas, really, because I think that's one that we really want to better understand. And speaking of that, what are some things that you hope to gain from some of the research that you're doing in order for us to better understand how the brain-gut axis, for instance, might affect adults and children?
So one of the well, there's two projects, I guess, that I wanna highlight here that I'm currently working on, both funded by the European Union. One of them is a big consortium project Europe-wide with basically all the big functional GI or disorders of gut-brain interaction groups being in there, where we look at mechanisms underlying comorbidities in IBS particularly. So both psychiatric and other somatic comorbidities, and we're looking at mechanisms across the entire gut-brain axis, starting at the microbiota and ending at the brain, or vice versa, to see how we can explain these comorbidities, which are the rule rather than the exception in these patients, I would say. And I am the PI at KU Leuven, leading the work package on brain mechanisms and stress physiology in that project, together with colleagues in Maastricht and in Gothenburg and in Barcelona who are involved in my work package as well. So that's one, I think, very important project, particularly in the context of IFFGD. Because we'll hopefully learn to understand these mechanisms better and try to hopefully better understand why some people suffer from all these comorbidities and others don't, which may be very important when it comes to maybe developing differential treatment for these comorbid or multi-comorbid patients. Then my second project is an individual project from the European Research Council. I just last December, I got a consolidator grant from the ERC, which is basically the biggest grant you can get as an individual researcher at my stage of my career in Europe. So I'm very excited about this. This is a more fundamental project where we are focusing on two important mechanisms by which the gut microbiota may impact stress responses and fear responses in humans, particularly being short-chain fatty acids, which are important bacterial metabolites that are formed when we eat dietary fiber-rich foods. And inflammation mechanisms as well, looking at how peripheral inflammation at the level of the gut or systemic inflammation can drive inflammation in the brain, and how that can impact the emotional brain and responses to stress and fear. So I think these are the two most important projects, not the only one, but definitely the most important projects that I have running at the moment. They both, well, the first one on comorbidities, the big consortium one, started last year, and the other one, the ERC one, still has to start. So I just got funded and we're working through all the paperwork. So that's going to start soon after I return to Leuven after my sabbatical here at Dartmouth at the end of April.
Yeah, and congratulations on receiving that grant, because like you said, that is very significant, and it's a testament to all of the research that you are doing in the field to help advance the field, for sure.
Yeah, very happy about it.
So we know that basically all research is really important because it really does help us understand these conditions and how it impacts the quality of life of all patients. But what would you consider being maybe your most successful research study?
So far?
Yeah.
Yeah. I've had many, so I have to think about it. Maybe I wanna. It may not have a lot of immediate implications, but I will start with a recent, fairly mechanistic study that I published a couple of months ago in Nature Communications, together with my colleagues here at Dartmouth, where I'm doing my sabbatical. And this is a study, a brain imaging study, where we looked at brain representations of different kinds of pain, including different kinds of gastrointestinal pain at the level of the esophagus, the stomach, the rectum, and we compared it with pain in other sites of the body. And we basically showed that there's a core pain processing system that actually, the response of which predicts how much pain people will feel, also for visceral pain, but we also found important differences between these types of pain. So if we're looking even within the gastrointestinal tract, there were important differences between the esophagus and the rectum, for instance. So I think to understand pain processing arising from the gastrointestinal tract better, we may need more fine-grained models, and we may have to distinguish different parts of the gastrointestinal tract, and different brain mechanisms may be at play, processing and generating pain from different areas of the gastrointestinal tract. And that kind of goes a little bit against the idea that yeah, all pain is more or less equal. So it seems to be a more nuanced story, where there certainly is some kind of common core pain system in the brain, but there's also particular mechanisms underlying different types of pain within the GI tract and across different body sites. So that's a fairly fundamental project, obviously, but we're kind of excited to take it forward and to start looking at patients as well, and see how these mechanisms or signatures or biomarkers at the level of the brain for different kinds of pain, how they may differ between patients and control. So that's kind of the next step we wanna take in that research.
Yeah.
So that's a fairly fundamental mechanistic type of study that we still have to take a step forward to patients. We're also looking at how the anticipation of pain and fear arising from the anticipation of pain, how that is basically encoded in the brain, and how the anticipatory response can predict how much pain people are gonna feel when they get a pain stimulus after the anticipation period. So that's one step ahead, and then the next step is to apply these models to patients and compare patients to control. So that's something that we're still working on. So I think that's something that I'm pretty excited about for now. And then obviously I've been involved in various more clinically oriented publications as well, particularly showing, often in collaboration with Magnus Simron in Gothenburg, with whom I'm collaborating closely, showing various ways how psychological processes are associated with increased symptom perception in patients with functional gastrointestinal disorders, IBS in particular. Also, for example, showing that increases in symptoms after a meal, that they are a lot more pronounced in people with psychological comorbidities, whereas people without these comorbidities actually don't seem to have a lot of increased symptom perception after meals. So that's more clinical work I'm quite excited about. Along the same lines, again in collaboration with Magnus, we started working on, and we're hoping to take it further, but a way to identify symptom triggers in people's diet. So to come up with a more fine-grained identification, a personalized identification of nutrient-related triggers for symptoms in patients with functional GI disorders, which I think will be an important step towards a more personalized dietary approach rather than the very broad one-size-fits-all dietary approach like the low FODMAP diet and things like that. So we're working on machine learning algorithms to try to predict symptom levels and symptom episodes from food symptom diaries. So that's something that is in the making. The algorithms have been written. They seem to work. We did some pilot studies that we published there, but we're currently looking into options to take that to the next step and validate that more extensively in well-characterized patient groups. So that's another thing that I'm pretty excited about, even though it's strictly speaking maybe not really a gut-brain axis project, but I think it's important.
Yeah, no, those are all very exciting research studies, and they will definitely be successful because being an organization that really does try to stand for the patient voice, I mean, patients always talk about the symptoms and how burdensome they are as far as living with gastrointestinal disorders, especially pain and especially the symptoms that they feel after eating. So those are all research studies that will be very impactful for the patient community. And how exciting for it to be coming from your lab.
Yeah, we're pretty happy about it too, and we see indeed that patients are excited about it because, of course, a lot of patients feel that foods or certain foods are an important trigger for their symptoms, but it's notoriously difficult for them and for doctors alike, I would say, to identify which kind of food is triggering which kind of symptom or symptoms in each individual patient. So we're looking into more data-driven ways to identify these relationships in a personalized manner, hopefully paving the way in a relatively simple way for personalized, more targeted dietary interventions.
Yeah, so exciting. We can't wait to see that in the near future here and the development of that. So you talked a little bit about some of the grant that you've received in order to do an independent study there, but what other acknowledgements have you received since winning the IFFGD award in 2013?
So yeah, I've mostly been focusing on getting funding. Of course, that's how it works when you're establishing your own group. I am very lucky to be embedded in a really large gastrointestinal research group at the University of Leuven, covering a very broad ground from very basic research on, for instance, the enteric nervous system, gut peptides, digestion and absorption, to more clinically oriented groups in both functional GI and IBD. So I am embedded in that, but of course I have my own niche because I'm a psychiatrist and I'm mostly interested in how the gut communicates with the brain. That's a pretty unique position that certainly helped me get a head start by joining colleagues' projects and now applying for also individual grants, which has really taken off over the past 2 years. I think we have been pretty successful. So I've mostly been focusing on grants.
Yeah.
Rather than awards. I would say I haven't applied for many, but I think certainly the award I got from IFFGD has helped me establish things and get those grants, because I often got, when I was looking at feedback on applications, of course, people appreciate that you get prizes and awards like that. Certainly my more clinically oriented work in GI or functional GI disorders has benefited a lot from that, I think.
Yeah. And funding is really important whenever we are talking about research. I mean, really, that's how you are able to kickstart some of the research that you are wanting to do and start answering some of those questions that many patients have. So funding is very crucial. So just the fact that you're applying for those grants, I mean, is a testament to all the research that you're able to do right now and that you're collaborating on with other people. So, what would you consider maybe being the most enjoyable part about being a researcher and why?
Oh, that's a relative. Well, there's many things, but I think it's an easy one in the sense that what I like the most is working with motivated, enthusiastic young people and training them to become the researchers of the future. And that's what really makes me happy. Of course, also the feeling that you're doing something useful, from which hopefully people will benefit. Even though I do a lot of fairly fundamental research without having a lot of immediate clinical implications, I do feel it's very important to understand these fundamental mechanisms better, before we can work on better clinical solutions. So I do get reward from understanding the fundamental mechanisms, but I get the most reward from working together with my team of really nice and motivated and smart and enthusiastic young people, that I can train and support to become independent researchers on their own in the future, hopefully. So that's what probably makes me the most happy about getting all these grants. Of course, it allows me to do nice research, but it also allows me to attract and keep promising young people to work with me together as a team, and deliver the best possible research. So I think that is one of the most rewarding aspects about being a researcher for me. And there's many other things. I do like the international aspect of it, the global collaboration, or at least attempts at global collaboration. Um, that's another aspect I really like. So there's many aspects, but I think the working with young, promising, enthusiastic, motivated people and try to help them make their way and become a good researcher themselves stands out for me.
Yeah, and I mean, the young investigators are really the hope for the future. So to
Yeah,
have mentors like you and other researchers is extremely
yeah, yeah,
useful and helpful for them.
yeah, yeah. And I'm in this, like, let's say, transition between being a junior and a senior researcher. I'm somewhere in the middle, I would say now. So also the consolidator grant from the ERC I got is aimed at these people, who are like, it's called consolidator for a reason. So there's three categories: a starting grant for really junior people, then there's the advanced grant for established researchers, and then there's this consolidator category for people between 7 and 12 years post PhD, to really consolidate their group that they are starting to establish, or have already established to a certain extent. So I'm in this transition period. So I see it a little bit as my task and my role to bridge the gap between the really younger generation and the more senior established investigators. And I'm happy to see that there's a, in my field, which is like the gut-brain axis, the link between psychology and gastroenterology, that kind of things. There's a growing community of enthusiastic young people, and I have to give the Rome Foundation, and particularly Laurie Kiefer, who has been the driving force behind this, credit for this initiative to establish a psychogastroenterology community, which is really taking off, particularly in the US. But I'm very happy that I attracted one of these people as a postdoc. So she transitioned from Laurie Kiefer's group at Northwestern, or Laurie's former group at Northwestern, I should say, to my group in Leuven, and she's really like trying to establish that kind of community now in Europe as well. So yeah, I'm very happy to see this young movement, let's say, in my field, and I'm trying to work with these young people and to support them in various ways.
Yeah, getting those fresh thoughts, and like what you were saying earlier too in the interview, it's not just something that's local to the United States. Like this is a global thing of collaboration between all of researchers. So that's pretty cool for you to kind of bring in that type of insight. So you talked a little bit about some of the things that you hope to gain an understanding about through your research studies, but what do you see as still being some of the gap needs in research, and how do you foresee some of those gap needs being fulfilled through the studies coming from your lab?
Yep. Well, there's many gaps when it comes to the gut-brain axis, right? I mean, it's getting a very, unlike when I started doing my research, interestingly, it's a very hot topic. The microbiota gut-brain axis as a whole. Not only I mean, the gut-brain axis was, when I started doing my research, was a small niche thing in a subgroup of people studying functional GI disorders. That was about it, right? Now it's the opposite. It's everywhere. Everyone's interested in the gut-brain axis. People studying neurodegenerative disorders, psychiatry, I mean, they're all knocking on my door and can we do this and this and that? So I have to be a little bit selective, because typically of an exciting new field in its infancy, there's also, I think the hype is a bit too big as well. So we have to find a balance there. So there's many gaps. I think when it comes to impact of the gut microbiota related signals on the brain in the context of stress related disorders, be it psychiatric or somatic. I think we need a lot more good translation of all the super exciting animal findings to humans. I think that is really lacking, and over-optimistic claims are being made about the translation of these findings. Lab animals are not humans, and when it comes to the gut-brain axis, that really applies even more strongly, because basically rodents are a pretty bad model for the entire gut-brain axis. Mm-hmm. Certainly when you're studying it in the context of psychology or psychological processes or psychiatric disorders. And their gut is different, their gut microbiota is totally different, and their brain and their behavior and their psychology is obviously also very different. So I see it a little bit as my task to try to do these early translation studies. And that's what that big ERC grant that I just got is exactly going to allow me to do. That's what I'm just telling you was basically the gist of that grant. So I made it very clear that there's like a lot of exciting work in animals, but it's being overinterpreted, that translation to humans has been very limited or poor or whatever until now, and that it's a very promising avenue, but we need better human translation at the fundamental level before jumping into all kinds of crazy wild guess type of interventions, particularly in patients. So that's I think a gap that needs to be addressed. When it comes to more clinical aspects of functional GI disorders or disorders of brain-gut interaction, I think what we really need is try to embrace the heterogeneity of these phenotypes or of these patient groups, and try to identify subgroups based on mechanisms, allowing us to have more targeted treatments for subgroups or individually tailored treatments. One of the examples I was giving earlier is the nutrition based work that we're doing, where we're looking at individualized nutrient-based predictors of symptom episodes in people, but it also applies to mechanisms. I'm pretty sure that there's going to be subgroups in whom the primary problem lies, let's say, maybe in the enteric nervous system. I have a colleague, he Boekstaans, who's doing a lot of nice mechanistic work on histamine and histamine receptors in the periphery, in the gut, or in the spinal cord. And that's nice, but that's not going to be the solution or the mechanism for all these patients. And I'm looking more at brain mechanisms, and that's also not going to be the problem in all the patients. There's going to be different subgroups. Just like in psychiatry, these are just their syndromes, which is a collection of symptoms. So people have similar symptoms. Well, maybe not even always, because some of them have diarrhea, some of them have constipation. So yeah, and there's probably going to be different underlying mechanisms playing a role in different people. And I think the big challenge for the future is to identify the subgroups and try to understand this heterogeneity better, to tailor treatments better to subgroups or to individuals, rather than sticking with these one-size-fits-all approach for the entire group of a given disorder of gut-brain interaction. So that's, I think, one of the challenges for the future, and with the nutrient-based work that I was alluding to earlier, and also the European consortium, where we look at mechanisms of comorbidity, we're trying to contribute to understanding that heterogeneity better, because I think it's a key thing to improve the care for these patients.
Yeah, no, and it sounds like, really, honestly, a lot of the research that is coming out of your lab will definitely fulfill some of these gap needs that you're speaking on. And we're so excited to have been able to catch up with you and just to really hear about some of the research studies in your lab and how you're moving it forward, moving the needle forward in advancing research and understanding of gastrointestinal disorders. So thank you so much for all of your unique insights in this topic. And thank you, everyone who has tuned in to our IFFGD Research Award Winner Series, where today we've had a chance to catch up with one of our past 2013 winners. So thank you, and thank you again for joining us today.
My pleasure. Thanks for having me, and thanks again for the award. As I mentioned earlier, it certainly helped my career, and it helped me get a head start when I was just becoming an independent researcher.
Well, thank you so much for sharing your story and for your unique insights on this topic.